mitochondrial DNA damage. All these things can really be directly attributed to the accumulation of lipofuscin. And so that's why it makes an exciting target for us, because we'd really like to see, gee, if we can keep this stuff down, if we can get rid of it,
right.
Where does that get us?
Sure. Now, in Louisville, you used the green pulsed laser on the worms, and it seemed as though the older worms who had a lot of pigmentation were becoming, basically they were being destroyed by the laser. Now,
Yeah.
how do you know, is this based on previous research with other laser treatments? I know lasers have been used in many medical and cosmetic procedures in the past. How do you know that your green laser with the worms that you were working with, that laser energy was being absorbed by lipofuscin and only lipofuscin, or perhaps was, is that laser light absorbed by other parts of the cell as well?
Right. Well, it does take some understanding of the existing literature and how this works. And based upon that, but also, you can look at our little experiment. It was kind of elegant because if an older animal is affected while a younger animal is not affected, then the only thing that can be different is what biological substances are different. What are in the old worms that weren't in the young worms? And so, you're limited to things like the fluorophores, things that give off fluorescence that accumulate with age. You're limited to oxidatively cross-linked and damaged molecules. Those are about the only kinds of things that you would even have to look at.
Okay. I see. Yeah, I guess that does make sense then. And what is the, what gives lipofuscin its pigment, and that particular part of lipofuscin that makes it absorb laser energy more than other parts of the cell? What's the part of lipofuscin that gives, just for my personal knowledge?
Sure. Well, the magic here is the fact that lipofuscin is dense. Now, within a living cell, we're not talking about bone here. We're not talking about anything. Sorry, I'm like in the middle of.
That's no problem.
Here,
no problem.
We're talking about something that is actually really dense. It's a cross-linked, compacted material. It's made up of all kinds of biomolecules and things from the cell that have gotten damaged, and they've started to accumulate in a dense little ball, kind of like a little piece of plastic or linoleum that's accumulated. And that makes it unique, because there's nothing else in the cell that is that large that's also that dense. And so those properties will give it it will give it a pigment that you'll be able to see under the microscope. That's how this stuff was first identified. We noticed there are these little black flecks in cells as they grow old, or in cells of an old organism. And actually they're reddish brown if you've got some good optics to look at it.
Reddish brown, is that due to iron? I've heard lipofuscin has a lot of iron in it.
Yes, rust, because you have oxidized iron. So that would give it a reddish brown color. And also it's got some fluorophores that depend on things like, I don't want to give specific biomolecules things, but
don't have to.
Depending on the lipofuscin you've got in what cell, you'll have different kinds of fluorescence, and it does have a fluorescence based upon the biomolecules you find in it. So I don't know where we start here, but that's exactly that.
Okay. Yeah, and I was just wondering, other people have thought about all the different advanced glycation end products and different junk that builds up within and outside of our cells throughout our life, and they've thought of different ways to clear this out, because this theory has been around a while that, hey, junk builds up within our cells, we end up with some damage, and we want to repair the damage or clear out the junk and help our bodies rejuvenate, our cells rejuvenate. Now, is the lipofuscin molecule, or the little dense packet of lipofuscin, are those real high energy chemical bonds that are typically not broken by enzymes within the human body? And do you think that the laser gives us a different angle that perhaps could be, break it apart? Sure,
you've hit the real central issue right there.
Okay.
And that's that. there's a reason this stuff cannot be broken down by the cell. The lysosome, where this lipofuscin accumulates, it has all kinds of enzymes that can break down just about any type of biomolecule you can imagine, and it regularly does. The problem is once you get some of these cross-links to form, you'll get these larger biomolecules that will get oxidatively damaged inside the lysosome, and they'll start to clump together. And you end up with it's really a physical problem where the enzyme, it operates in this quasi-solution that's in the lysosome. All of a sudden it can't get at these bonds, these inner cross-links between these larger molecules.
Oh, okay.
And so it's really a physical problem of access. All of a sudden the enzymes, they can be there, and they are there in great quantities, but they can't do their job. They can't work. So
all right.
The laser enables us to just flat out thermally heat this material so quickly that it will disintegrate.
It just and that's basically what happens. It absorbs a lot of energy, and basically it heats up, and these bonds break, and then we end up with smaller, digestible pieces of lipofuscin then, or it just breaks up into something else?
You'll end up with complete carbonation if you use carbonization, if you use a high enough power.
Oh.
Now, of course, once you do that, you basically completely destroy the cell that contained the lipofuscin and probably some neighboring cells.
Oh.
So also, yes, you can break it down into smaller chunks. One thing they've seen with melanin in some of these studies is that you'll see some fragmentation of melanin granules where the laser was hitting them, on the side the laser was hitting them. And that's, so yeah, you can end up with smaller pieces. Now, of course, we're not sure about exactly how that all works out, but we know from some existing experiments in the literature that it works. With at least certain cell types, you can get rid of the lipofuscin in the tissue and kind of reset the clock.
Okay. Now, I just want to mention we have got a viewer question, and I want to remind all the viewers here tonight that if you do have any questions, feel free to ask them as we go along in the interview. We'll be having the interview another 10 or 20 minutes. And the first question I have from an audience member, kind of an interesting one here. I'm not sure if you have data on this. Are there any major differences in the effects of lipofuscin accumulation between different species?
Actually, well, of course, there's such tremendous variety in biology. By the time you get down to simple organisms, simple multicellular organisms like sponges and hydras, you're at a type of organism where all the cells are replaced on a regular basis. They continually divide and rejuvenate the organism. Those kind of organisms don't accumulate lipofuscin at all.
Really?
What's interesting is, exactly, and well, neither do cancer cells. You can have a tumor. A tumor, if it grew for 100 years, would never accumulate lipofuscin because it's continually dividing the cells and diluting its contents.
Wow. Okay.
So there is such a thing as immortality in nature of the sort, and that's encouraging. But for more complex organisms like us, and even like the nematode worms we look at, once you get complex enough that you need a circulatory system or nervous system of any sort of complexity, you need what's called post-mitotic cells. You need cells that can be laid down as the organism develops, and they basically lay out a structure that performs a given function. And they can't be moving around a lot, and it's very difficult to reorganize them if things get disrupted.
Right.
Those are the types of organisms in which we see this lipofuscin accumulate. And in those types of organisms, from worms to mice to cats and dogs to humans on up, it's the same. You see exactly the same rate of accumulation of lipofuscin relative to the lifespan of the organism.
Okay.
So if it's about 3/4 of the way to where they typically die off, you'll see about the same amount of lipofuscin in a worm versus a human who's about 3/4 of the way to the average age of death.
Okay. Yeah, and now speaking of worms again, could we move on to what is proposed with the research grant that the Institute is going to raise some money for. And what I've read so far, the preliminary little description is that you'll be performing some lifespan tests on worms, and also you'll be testing the